Welcome to this explainer. Look, as future clinicians, you're going to be constantly navigating the really delicate balance of human homeostasis. Today, we're zooming way in to focus on the literal foundation of that balance, protein folding, and more importantly, its pathological consequences. We're going to look at how a simple linear sequence of molecules transforms into the incredibly complex machinery of life, and exactly what happens to your patients when that machinery breaks down. Here's our quick roadmap for today. We'll hit 1. The architecture of proteins. 2. Form dictates biological function. 3. Regulating metabolic enzyme activity. 4. When protein folding fails. And finally, 5. Clinical impact of misfolding.
All right, let's jump right into section 1, the architecture of proteins, moving from amino acid sequences to 3D conformations. Think of this like building a house. A linear sequence of amino acids is really just the starting line. Held together by peptide bonds, this primary structure starts folding into a secondary structure, forming your classic alpha helices or beta sheets, which are stabilized by hydrogen bonds. From there, we advance to the tertiary structure, which is that complex 3D folding driven by R-group interactions. And finally, for some proteins, multiple subunits actually assemble together into a quaternary structure to form these massive cooperative complexes. Now, why do they fold this way? Well, it's all driven by highly specific biochemical forces. We're talking hydrophobic interactions that push nonpolar chains right into the core, alongside electrostatic bonds, hydrogen bonds, and covalent disulfide bridges. These aren't just random. They are absolute prerequisites for biological activity. Literally, without these specific forces stabilizing that tertiary structure, the protein is completely useless to the cell. Moving on to section 2, form dictates biological function. Let's look at the molecular building blocks of life. It's honestly amazing how specific protein classes rely entirely on their intricate architectures to execute highly specialized physiological and metabolic jobs. For example, structural proteins like collagen and keratin, they're tough, they're insoluble, perfectly built for mechanical stability. But contrast that with enzymes like DNA polymerase, which are globular and incredibly reactive biological catalysts. Then you've got hormones like growth hormone regulating huge physiological processes, and transport proteins like hemoglobin, which are specifically engineered with a quaternary structure to neatly bind, transport, and release oxygen in the blood. Form absolutely dictates function here. Which brings us to section 3, regulating metabolic enzyme activity, the dynamic machinery of the cell. So the key takeaway here is that these aren't just static building blocks. When properly folded into its active conformation, an enzyme has a highly specific active site that drastically reduces the activation energy of a reaction. But obviously, to prevent total metabolic chaos, the cell needs an off switch. Enter structural flexibility and allosteric regulation. Basically, a molecule binds to a totally different site on the enzyme, physically shifting its 3D conformation. The active site changes shape, and boom, the enzyme is inactive. It acts as this perfectly timed instantaneous metabolic off switch. We've spent some time on the absolute brilliance of healthy physiology, but now we really have to ask the critical question that shifts us directly into clinical pathology. What happens when this entire intricate folding process just fails? Let's look at section 4, when protein folding fails, the precarity of molecular architecture. Inside every single cell, there is a high-stakes battle going on. Molecular chaperones are basically acting as cellular guardians. They bind to forming polypeptides, assist in correct folding, and actively prevent premature aggregation. But what if the cellular environment changes, or mutations pop up? Well, those chaperones can get completely overwhelmed, and the result is pathological aggregation. These misfolded proteins expose their sticky hydrophobic cores, they start clumping together, and they entirely lose their biological function. So when physiological folding fails, or say, when external toxins hijack the cellular machinery, the immediate result is profound cytotoxicity and metabolic dysfunction. The cell totally loses the specialized function of that protein. The misfolded aggregates physically block cellular transport, and delicate metabolic pathways are just completely disrupted. Plus, external threats, like diphtheria toxin, for example, specifically target the protein synthesis machinery itself, creating massive, severe cytotoxicity that you're going to have to manage clinically. And finally, section 5, the clinical impact of misfolding, going from microscopic flaws to macroscopic disease.
The failure to maintain a stable 3D conformation directly translates to the severe, life-altering clinical pathologies that routinely present in neurology clinics. Think about Alzheimer's with amyloid beta plaques or Parkinson's with alpha-synuclein aggregates. You guys, these aren't just abstract textbook concepts. These are the microscopic misfoldings that gradually destroy memory, motor control, and ultimately your patients' lives.
There's a great insight here highlighting that understanding protein structure provides essential insights not only into how proteins function, but also into how they can be manipulated for biomedical applications and targeted therapies. And it's so true. As structural biology advances, especially with incredible tools like cryo-electron microscopy and AI mapping 3D conformations from primary sequences, we're entering a totally new era of medicine. We aren't just observing misfolded proteins anymore. We're actively working on designing targeted therapies to stabilize them, clear them out, or just prevent that aggregation in the first place. So I want to leave you with this final provocative thought. Will decoding the structural vulnerabilities of misfolded proteins be the ultimate key to future drug discovery and actually reversing neurodegenerative diseases? Understanding how proteins fold, function, and fail isn't just something to memorize for your boards. It is literally the frontier of modern medicine. By grasping these mechanisms right now, you're equipping yourself to understand the targeted therapies that might one day cure what we currently consider incurable. Thanks for joining me on this explainer, and stay curious.